Kumar sir, over to you, sir.
Yeah. A very good morning to each one of you. It's indeed my pleasure to invite each one of you on behalf of the management and staff of Sri Vishnu College of Pharmacy. Today is day 4, and the first session is by Dr. Varma Manthana. And it's indeed my pleasure to see Dr. Varma today on live. He actually belongs to this place called Bhimavaram. And it's a very proud moment for us to see people from Bhimavaram in the top positions in companies like Pfizer. And Dr. Varma is MS and PhD from NIPER, and is followed by postdoc from University of Minnesota, and is presently working with Pfizer. And R&D, I'm also following his research work through LinkedIn. I'm quite impressed by his progress and achievements, and the topic that he has taken is gut instincts. I'm sure everyone should have instinct to see his gut feelings on this topic. This is very relevant and most commonly used route of administration, and almost 40% of drugs in the market are through this route. And understanding that oral absorption of the molecules is very exciting. In fact, many of you maybe know Dr. Lipinski rule that also came from the data analysis of the Pfizer data. So today is yet another start from Pfizer, and Dr. Varma, and he's going to enlighten us with his talk. So I really thank Dr. Varma for accepting our invitation, and it is night 11 o'clock or so, and he's trying to keep himself awake and make us enlighten us with his rich knowledge, and his publication record is so great, and contribution to the field is so good. I'm really proud of him as a NIPER alumni. And with this note, I also thank all the speakers of the past sessions who made this program highly successful, and looking forward. And I believe also that today's session is equally exciting, probably even more than the before. And the question and answer session is also going well. I request each participant to note down your questions and ask at the end of the session. Please make use of this opportunity. These are all rich knowledge and gained through practice, not by reading books like us in academic institutes. So you really get the good insights, and please make use of this opportunity. So with this note, I really thank all the previous speakers. At the same time, Dr. Varma for accepting our request and giving his talk today. So with this, I'll ask Dr. Ravi Kumar to formally introduce today's speaker.
Good morning, one and all. I'm Dr. Ravi Kumar, Associate Professor, Sri Vishnu College of Pharmacy. So I'm delighted to welcoming you all for the 4th consecutive day of the AICTE sponsored STTP on advances in pharmaceutical formulations and regulatory pathways globally for small molecules, medical devices and biologics, conducted by Sri Vishnu College of Pharmacy, Bhimavaram. So it is my privilege to introduce the first speaker of the session to the participants, that is Dr. Manthana Varma. So Dr. Manthana Varma is currently working as a research fellow at Pfizer USA. So Dr. Varma received his B.Pharm degree from Kakatiya University and MS degree and PhD from NIPER Mohali, Punjab. Later, Dr. Varma moved as a postdoctoral fellow at the Department of Pharmaceutics, University of Minnesota. In 2008, he joined Worldwide R&D Pfizer, Groton City, US. And Dr. Varma holds an adjunct faculty position in the Department of Pharmacy of University of Rhode Island. His research is focused in the field
Hello.
So sorry, sir. I think there is some technical issue with our end.
No problem. No problem.
Just 2 minutes, sir. 2 minutes.
No problem. Yeah.
So sorry for interruption. I'm almost done with the speaker profile. So Dr. Verma supported preclinical and clinical development of several Pfizer compounds in the oncology, diabetes, and more recently in non-alcoholic steatohepatitis, that is NASH, NAFLD therapeutic areas. So he published over 100 original articles, reviews, book chapters, and presented over 75 presentations at the conferences in these scientific areas. So now I take this opportunity to invite Dr. Verma to start the session. So session is over to you.
Thank you, Dr. Ravi Kumar. I just want to check if you can hear me clear.
Yes.
Okay,
Yes.
perfect. Give me a second before I can share my screen. Okay. Can you see all right?
Yeah.
Yes.
Okay, perfect. Yeah, thank you, Dr. Ravi Kumar, for that elaborate introduction. I am indeed grateful to be here presenting some of our stuff. Thanks, Dr. Nivani, for having me talk on this topic, as well as reaching out to a wider scientific community in India. I'm always looking forward for those kind of opportunities. So this is perfect. And also thank to Sri Vishnu College and its management for making this happening. It's a wonderful event. I have been following it for the last 2-3 days. So a lot of good stuff going on here. So the topic I picked for today is kind of talking about the oral absorption from a drug discovery perspective. More importantly, talking about the physiology of the gut and how that impacts the absorption, and why do we need to know this is one of the aspects that I would be covering. So technically, the goal for our talk today is to understand the drivers of the absorption. We wanted to understand this so that we can assess the risk of finding of screening out the bad compounds so that we can enable the design and selection of right candidate that can go into the clinical development. Obviously, we are going to put these compounds first time in the human, so we wanted to get it right based on the experiences or based on our physiological based understanding of this process. So with that, I just wanted to, show a couple of pictures of where I am now. This is the place, Groton, Connecticut. It's a beautiful town. If you visit East Coast, you should stop by. It's a nice beach town. As you can see, one of the popular things about Groton is it's the first submarine place station of the world. It's called the capital of the world for submarines, and you can see one of those there. And the submarine station is one of the larger employer in the area. After that comes Pfizer. So this is the campus of Pfizer, and you can see it's again close to the water, and it's a beautiful site. So yeah, it's been a while I have been there because of the COVID-19. It's almost like since March, most of us have not gone to office. I hope it is as it is when we go back. With that, let's go into the topic. I come from a background of drug discovery, so I emphasize more on how the discovery process works and what are all the processes involved, or what are all the activities involved in identifying the compound that has the probability of success in the clinic and eventually gets into the market and makes cures some of the diseases for the patients. And the goal always here is the patients come first, and we are always working towards a therapy to offer them the right options and in managing some of the diseases. So some of this. So what I'm going to do is a couple of slides, just give a very high level overview, and I understand this might be basic information for some of you. Just I appreciate the tolerance at that point. But just give me a few seconds here while I look into some of these aspects for you guys. So drug discovery, it's a team effort. It involves multiple disciplines. We're talking about various groups coming up with ideas and then synthesize a whole set of compounds, which will be screened for various processes, including the ADME, which is the absorption, disposition, metabolism, and clearance aspects, which is the topic that we will be covering today. Obviously, there's pharmacology, toxicology, and other aspects that goes into that, and I'm going to touch a bit on that. So compound selections based on a whole lot of experimentation that happens in this level. We eventually pick one compound which has the highest probability to make it into the market, which eventually gets through the phases of clinical studies, and ultimately to the registration, regulatory authorities, and other aspects of it. So that's a big picture, as most of you working in the drug discovery knows, drug discovery development or any other supporting groups are well familiar with that. So what is a drug design team technically? There is a lot to it. Again, it's a complex network of activities, and I think I'm going to just show you like a subway map in the next slide. But high level, a team comes up with an idea based on the existing literature or existing knowledge on the target identification, and then comes up with a structure or a lead molecule or some series which has the potential of getting a good efficacy and ultimately producing a good activity for that target, right? So there's teams like chemistry, pharmacology, ADME, which is PK in other terms, toxicology. This is an highly interactive teams working with each other and identifying or optimizing these chemical structures in such a way that we identify a compound which has all the right properties in these individual departments, which can eventually make it up into the clinic. So this is the kind of activities that's happening at the drug design phase, and I'm part of some of these activities supporting the ADME aspects out of it. So this is the subway map I was talking about, right? The drug discovery development involves a lot of groups, and some of this stuff perhaps I do is kind of a drop in an ocean, but whatever we are doing or contributing to these individual components still is a big deal, and it is still an important piece of the puzzle, which drives the identification of the right compound into the market, into the making it to the market. As I said, the drug design process involves a tiny bit of this, which I showed in the previous slide. But as I said, that's the starting point of an API, which will eventually be there in the market if it is successful pretty much forever, right? So, those kind of activities is as critical as anything in the rest of the map here. All right, so what are the what are we interested in from a drug design perspective? And since we are going to talk about pharmacokinetics or oral exposure, excuse me, our goal, our ultimate goal here is to come up with a molecule which is pharmacologically efficacious. At the same time, there is tox endpoints and other stuff that we will not talk about, but the compound has to make into the near the target and show, therefore eventually causes the pharmacology, right? So it's just a simple equation that defines the dose. Dose is everything in the drug discovery, and the dose is nothing but as a component of pharmacology, which is the efficacious concentration, and the concentration or the exposure that compound can reach at the target site. So when you take the pharmacology part outside this equation, our major focus will be on the pharmacokinetic perspective, right? Which is defined based on the clearance, the tau value, which is the dosing interval, and then the fraction absorbed or the bioavailability. Technically, it's whatever of what fraction of the dose actually gets into the systemic circulation. And we will be talking obviously heavily on these components. So to ensure the compound to be safe and efficacious, these are again the primary requisites, and this is what we are optimizing from the lead to the candidate selection and eventually into the NCE. So why is this important? Why are these multiple components important? So this is some of the analysis that was done by the Pfizer based on the successes of phase 2, phase 3, and compounds that are reaching phase 2, phase 3, and eventually making it to the market. So what are the important components of those success stories? When Pfizer has done quite a bit of analysis using 44 phase 2 programs, we identified that the compounds or the NCEs with a good exposure and a good pharmacology actually has the higher probability to reach to the market. And this is pretty intuitive, but it's always good to have that holistic data set, which really talks in the perspective of numbers. And based on that, within the Pfizer organization, we have put out something called as a 3-pillar approach, where we need to make sure we get the right exposure, we get the right target binding, and once you have the target binding, that has to translate into a good functional activity, which is the pharmacological activity. Unless you achieve at least 2 out of the 3 pillars, it is very less likely that the compound can make it past phase 2 or phase 3, which eventually means a failure at a later stage is not a good aspect. All right, so I think I led this discussion from the multiple components of identifying an optimum compound to the exposure and pharmacology. And for the sake of it, we'll keep the pharmacology apart. That's a topic for a different day, and I'm sure a lot of other speakers have spoke to that in the past. So my focus will be on the absorption part, as the title speaks to. So when it comes to the absorption, we are again focused on the oral absorption. Most of the programs in the small molecule therapeutic area, at least from Pfizer, is predominantly oral. And there's good reasons for that, and I don't need to speak to that within this community, and you're very familiar with that stuff. When you take a tablet, and again, this is very basic, just bear with me here. There is a solubility component, permeability, metabolic stability, and once the compound makes through, or the drug makes through these elimination processes, it will end up in the circulation, which is what we are interested in. This is the PK profile which we are interested in, and we are always working towards a PK profile that gives you exposure well above the concentration that is needed for the efficacy. That's our primary goal. And we need to identify a compound that actually reaches that goal. Sometimes we can work on fixing the solubility and other stuff, or maybe formulation approaches, but you always need to have an API that has those intrinsic abilities to really produce that exposure, which is well above the concentrations that is needed for the pharmacology. So let's narrow this down a little bit to the solubility permeability components. So this is a very high level picture, right? Let's try to dig into more details and talk about these individual components and say when you're in a discovery stage, what are those components that you try to optimize so that you maximize the exposure that you needed in the target side. Also try to minimize the dose as much as you possibly can. That should be your goal as in a drug discovery setting. So, so when you talk about the details of what is happening to this API or the formulation that we are dosing, it involves multiple steps. There is the release from the particulate material or just the solubility of the compound within the gut or individual segments of the intestine. And once it releases, it may achieve supersaturation that is defined by the solubility, and eventually it could get precipitated. Or it can actually also interact with some of the components in the gut, like bile acids and other stuff, forming complexes and other stuff. So the reason I'm bringing it up is like, so there are multiple arrows to this and multiple activities going on simultaneously. And these are all important components which ultimately defines what actually goes into the blood compartment. So that's the solubility piece. And then once the compound is available in solution, it has to cross through the biological barrier. We call these enterocytes. Enterocytes is the epithelial cells that line the gut wall, and the compounds has to go through that membrane, and we'll talk about a little bit on that in a little more detail. So let's try to split this into two pieces and talk about it, right? The first piece is the solubility, and the second piece is the permeability. We'll talk about it a few slides later. Solubility, as you all know, it's the concentration of the solute in its highest saturated form. And it is defined by the conditions of the media, which is temperature and pH in this case. And the reason we only talk about temperature and pH here, because that's what we are more interested in the physiological sense. But one could argue also there are other components in the gut which could also influence this. And that's a topic for a different day, which is like the biorelevant dissolutions and other stuff. I'm not going to touch upon that. So solubility and permeability. There's something called biopharmaceutics classification system. I'm sure a lot of people working in pharmaceutics are pretty familiar with this. Maybe pharmacology and toxicology folks are not very familiar. So let me give you a high level overview of BCS. BCS is a classification or a grid which is based on the solubility and the permeability axes. So a compound with a high solubility and high permeability typically considered to have very good absorption. So you should technically not worry about absorption of these compounds. They're highly soluble, they're available for absorption, they're highly permeable. That means they can easily cross through the membrane. The second class is low solubility, high permeability. That means barrier is not an issue, but the solubility can be an issue. And this is the class we often see. You know, high solubility, high permeability is not. It's something that we always aim for, but it's not necessarily realistic in a lot of therapeutic areas. So we often see classes like class 2 and class 4, but the solubility is one of the major limitations. There's also class 3 where the solubility is good, but the compound is either very hydrophilic or it does not have the right properties to cross the membrane. Therefore, even they are good, highly soluble, their absorption is going to be low. And we don't want to get into that space because there's nothing from a formulation perspective we can manipulate that and fix those. So whatever you have to do is, in this case, you always want to push by chemical design, try to modulate the chemistry so that this moves towards class 1 space, so that you at least have a control on how you formulate that and maximize the exposure again. Okay, so we'll also talk about some of the efflux transporters later. So our goal, as I said, is always to reach high solubility, high permeability, but that's not often realistic. But you know, sometimes when you're in the borderline of those classes of high solubility, high permeability, or low solubility, low permeability, or if you have one of the components to be a decent value, you may still see a good absorption, and you probably don't need to worry too much of not able to reach 100% bioavailability, whereas you might get like a 70 or 80%, which is still a good amount to probably trigger a target or something like that. So that is what you're seeing here in this slide. This is a chemical series that was run in Pfizer. It's an antimicrobial agent series. So what it has, like this is way before my time when I joined Pfizer. So what you're seeing here is a series of 27 compounds where they measured the solubility and permeability, and then also looked at the rad bioavailability for these compounds. And you can see the compounds with low solubility, low permeability has a very low bioavailability, whereas the compounds which are high solubility, high permeability gives you a very good absorption. At the same time, you can see those squares here also in the borderline. The squares imply high absorption rates. You can still see those high absorptions in the borderline cases, which means, sometimes when you're in the borderline, you don't kill yourself to, push it to a better position. You probably are in a decent shape. You further wanted to test it to make sure it has enough legs to move into the next phase of the discovery cycle. But overall, this chemical series story really describes you the importance of considering this solubility permeability component from a BCS perspective, so that you can identify the right compounds or the right series which can have a good absorption component. So I don't want to get into too many details here. We are not talking about formulations. I knew in this webinar series, a couple of speakers have spoken about the formulations and dissolution components in nanotechnology. The major point I wanted to make here is all these processes of disintegration, dissolution, and eventually precipitation and other stuff. It also depends on the API. It actually depends quite a bit on the API, right? Like the physchem properties of the API. One of the key components is the intrinsic solubility, which defines how much of a compound can be solubilized. And there's molecular properties like lipophilicity and other stuff that defines it. Probably we will not go into details of that, but obviously any property of an API is defined by its physicochemical properties, as you all know. The point here is, what are those key properties that we should be after in order to better understand if there is a limitation, and also translate that data when we move forward. So it's pretty common in the drug discovery programs to optimize the chemical structures to enhance the solubility. Because as I said, often we start with a lead compound, which is more like a brick-like, but then we kind of optimize, the drug discovery programs kind of optimize it to do their best, so that once it goes through the preclinical stage, it should be much easier to pass through the clinical program. We don't need to come up with the fancy formulations to fix some of the solubility limitations, right? So it's reasonably obvious here. So try to fix the molecule as much as possible early on, and that's the biggest part of the design optimization. So what are the ways a chemist or a design team approaches solubility? It again is a manipulation of the physicochemical properties, primarily trying to ionize the molecule so that the compound is ionized at a given pH and produces the solubility. Lipophilicity is another thing, hydrogen bonding and other stuff. There's a lot of SAR around it. And you can see some of the examples here. For example, this R group on this pyridine group, if that R group was changed from quinoline to phenyl, the solubility has gone from almost 0 to 160 micromolar. 160 micromolar may not be a super high solubility, but it's good enough for the compound to reach a decent exposure and has the ability to move forward. So there's another example. There's tons of examples out there. You can look through the literature on some of these case examples. In this case, this benzamide group has been functionally modified with a methyl, and the solubility goes up it has a very big value change, which is the great thing, right? This, like, fixing that small methyl group has a huge impact on the solubility. What else do you ask in this case? And that too without compromising too much on the efficacy. In this case, it's a CDK2 inhibition. You might know about this classical example of indinavir. Indinavir again is when the original lead compound came up, which showed a very good efficacy for the HIV. It technically had no bioavailability, and then the optimization process of this molecule led to addition of an ionization center, which eventually led to a 60% bioavailability, right? The solubility went almost from 0 to something. The something does not need to be super high. It needs to be sufficient so that you get a decent exposure in the human, and this is now a successful drug. So this is a classical example. Again, some of these old stories, but the good stories to talk about. So once we talk about the intrinsic solubility, there's a lot of physiological components that one has to factor in. And one of the primary things that we consider is the pH of the gut intestine or the intestinal tract. And the intestinal tract has a gradient of pH. It starts from stomach of 1.5 and goes all the way up to 8.5. And the important thing to note is this pH can change as a function of other components like the fasted versus fed state. That means if you take a pill, fasted versus a fed state, you might get a different solubility for a basic drug and therefore get a different profile. And excuse me, we'll talk a little bit about some of that stuff. So considerations to some of these physiological components is pretty critical when you're actually making those final predictions of FA. So the proton pump inhibitor effect is pretty classic if you think about it. So a lot of proton pump inhibitors that is pretty widely prescribed has a huge impact on the gastric pH. And I'm just showing an example here of change in the gastric pH when you dose esomeprazole, which is Nexium. And it dramatically increases the stomach pH, which means a basic compound, a weakly basic compound, which has to have an acidic pH to have a high solubility, now does not have that milieu to produce that high concentrations. That could lead into changes in the exposure. So this is a classical study where you see when you dose this weakly basic compound along with Coca-Cola versus water versus a packet of esomeprazole, you will see a very different profile. And this is primarily driven by the solubility that is occurring in the stomach, because as you see the profiles here, in the presence of a PPI, the stomach pH is so high that the solubility does not reach enough levels. Therefore, you have typically a low exposure. Versus when you drink a carbonated water along with it, it shows you a very different exposure. So the same molecule or a same formulation, same drug produces, three highly variable profiles based on how the patient is taking that formulation. And those are the important components when you talk about this. And this is a pretty common phenomenon if you think about it. And I just listed up drugs, primarily the tyrosine kinase inhibitors, which are like the hotcakes these days, particularly the oncology and also inflammation therapeutic area. And you see some of the blockbuster drugs have been impacted by taking these different kind of PPIs, and one needs to better understand that so that you can essentially, design uh, or design the label and other stuff for the specific population, and try to uh, provide this information in the right format for the patients. So, and again, this example or this slide demonstrates how important these physiological components are in the overall exposure that you're looking into, which eventually will obviously translate into efficacy or other adverse activities and other stuff. Can we predict the effect of PPIs and other stuff? We probably can to some degree, and obviously it depends upon complex multifactorial activities, the solubility, pH-dependent solubility, how high is the intrinsic solubility, the other factors like chloride ions, and some of the stuff that is openly circulating, openly flowing within the gut may also have some interactions. And obviously there's a lot of other like the solid state related details, like whether the precipitation forms crystals, what would be those size of those nanoparticles and those kind of stuff. So without getting into all of that details, again, I'm not a formulation scientist, so I don't want to get into those specific details, but you can think about the intertanglement of these multiple factors that could contribute to that effect. So you may not see that effect for all the drugs, but you will see for some of the drugs. And these are some of the reasons why you see that. It's good to predict that upfront. So there are complex models to do that. But at the same time, the confidence in these models are still, in the process of learning, and it could take some more time to get a better handle of that. So this is some of the exercise that we did, trying to understand what are the fundamental properties of a molecule that is causing this effect of PPI. For example, what you're seeing here is the change in exposure of a basic drug in the presence of PPI versus taking along with the water, which is the control. And you can see the exposure goes down as a function of pKa. If the pKa goes down, the exposure is also going down. And again, this is a limited set of compounds, which is specifically for a set of TKIs. But you can see a trend. But at the same time, these compounds also have low intrinsic solubility. So it's probably a combination of that intrinsic solubility and the pKa that is driving the larger effect of, for example, if you take this compound with a PPI, you're technically having only 30% exposure of what you should typically have, which is probably leading to subtherapeutic efficacy and other stuff. The other components that could contribute to the variable oral absorption, and again, there is some of these things you cannot really manage in the discovery programs. This is information that is good to have when you move towards the development and when you're co-dosing with other drugs. Some of the drugs, particularly the GLP receptor agonists, can actually delay the gastric emptying, which can lead to, if you're delaying the gastric emptying, we are essentially pushing the Tmax to a later time and essentially suppressing the Cmax. And that's what we see with a lot of this GPR, GLP-1 receptor agonists. And this is the list of compounds that actually show, does this effect, is this effect clinically meaningful? Probably not with some of these drugs like atorvastatin, because all you're seeing is a suppression of Cmax and the exposure still remains the same. But this is just to demonstrate on the effect of these physiological changes on the absorption of some of these drugs, which takes relatively more time to absorb and other stuff. Finally, age, it's not about the influence of other drugs or on this gut physiology. It's important to consider the patient group, and this is a chart that is talking about the acid secretion as a function of age. That means the older people have a much lesser acid secretion, which means the gastric pH in the geriatric population, which often takes a lot of these drugs, is higher, which is probably relating to what I have showed in the case of esomeprazole example, right? You're reducing the you're increasing the gastric pH, which means you're reducing the solubility, and therefore you're probably getting low exposure for weekly basic drugs. As also there is an increase in gastric emptying time, which is again an example I talked about the GLP-1 agonists. Naturally, the intestinal motility changes. Also, unfortunately, the absorption surface also changes. So all these components contribute to some of these variabilities that we are seeing from one population to the other. Although we do a lot of developmental studies in healthy volunteers, our target population who is taking the medicines in the real world is probably towards the higher age groups. Therefore, it's important to factor these when we do these mechanistic models. Therefore, we better understand the exposure in those populations too. So let's switch gears here from the solubility to permeability. Again, I covered quite a bit of high level stuff here. We can always talk about details, but given the diversity of expertise in this call, I just wanted to touch on the high levels, and obviously we can follow up on some of these in the discussion and question and answer session. Permeability. Permeability is essentially a drug which is in its soluble state, crosses through the membrane, and there are multiple mechanisms through which a drug can cross. The paracellular, transcellular, compound can utilize some of the uptake transporters on the cell membrane, and we'll talk a little bit about that later. And the compound can be limited or can be effluxed out by efflux pumps, which will actually essentially reduce the overall absorption. And there are endocytosis processes also involved in the gut absorption, but this is more like particulate material when you're talking about probably some nanoparticles and other stuff. We are not going to talk about that because we are only focused on the API or the new molecular entity per se. So design optimization, technically essentially trying to look at the structure, chemical structure, and talk based on the past learnings, what are the functional groups that one could alter in order to maximize the absorption. And again, we are talking about absorption here. There's also a lot of other optimization processes from the efficacy, toxicology perspective and other stuff, right? But as a drug design team, as an ADME scientist, there is always inputs we throw towards a structure and try to challenge it by saying, if you modify this group, probably we'll get a better solubility, later permeability. And I talked about these solubility examples. When it comes to permeability, I think the important components that has to sit through is the size, hydrogen bonding, and lipophilicity. And one has to also think about the what could transporters do to this overall effect. So it's important to think that through in certain cases, not necessarily in all cases. So how do we know that the size, hydrogen bonding, and lipophilicity are important components in the permeability. And that goes to the classic rule of 5. This is the work that's been done like probably 25, 30 years ago by Chris Lifinski, who's one of the senior most chemists at Pfizer. He was also at Groton all his career. So he came up with something called rule of 5, which essentially means if we have all these properties in a molecule, the probability of the compound having high bioavailability is very high. And these properties are the compound lipophilicity or log P below 5, molecular weight below 500, hydrogen bonding ability below 5 or 10 based on whether it's a donor ability or an acceptor ability. I'm sure a lot of you have heard about this, but just if you're not familiar with this, just digest this for a moment, right? This is based on an analysis of more than 1,000 compounds, and a typical small molecule usually have these properties unless you're talking about some of these high molecular weight compounds like the HCV, HIV drugs, which are typically towards 700, 800. They're often challenging. So within these properties, you always have, you often have a good absorption, and that's an important component to think of. But at the same time, you will always see an outlier where, within these properties, you're still not happy with what it is producing. So we did a lot of analysis, subsequent analysis on looking at the PhysChem properties, on whether we can differentiate some of these physicochemical properties and link it to the bioavailability. So this is a part of the analysis, very high level, and you can see there is definitely a trend. If you increase the molecular weight, the bioavailability is going down. So there is definitely a trend, like similarly with the lipophilicity should be optimally below 5. You don't want super high number of rotatable bonds because they're detrimental to the absorption. Polar surface area, there's also a trend here. But you also see like those walls of data points, which kind of reflects, although the mean and median values shows a good trend, you will always see a huge number of outliers. And you may actually have very good property for those PhysChem properties, but your compound may be somewhere here. So you have to be a little careful about that. And this is where looking at an individual property may not really make sense, may not really translate. And this is where the quantitative SAR models really help in building up those confidence. However, if you try to link this up from the rule of 5 perspective, and this is what you are seeing, the median fraction absorbed. Based on the number of violations, you will have very high absorption when there is no violations. That means your compound fits very well with the rule of 5. But if the number of violations is high, that means your molecular weight is more than 500, but rest of the 3 properties fit within the criteria, you still not bad. But the problem is when you exceed the criteria for all the properties, that's when it's a big problem. It's technically a no. At least the Pfizer philosophy is if your rule of 5 violations or more than 1, you may want to go back to the design board and try to come up with a new series or a new chemical to try to fix that, because we cannot risk to go into a clinic with suboptimal compounds like that. And then there are in vitro tools. Naturally, we are always not necessarily relying on the trends or the quantitative SAR models. You always need to test it either using in vitro and in vivo tools. There's a whole bunch of in vivo tools that we will not talk about, but this is only one slide that I talk about on the in vitro tools. This is called a Transwell assay. I'm sure a lot of you are familiar with this. Technically, we grow cells on top of it. It could be Caco2 or it could be specific transfect cell lines. And once you grow the cells for a few days, you are able to now do an experiment from one side to the other side. From top to down typically is called apical to basolateral, which is, if you put this in a physiological sense, this is your lumen versus towards the blood compartment. And the good part with in vitro studies is you can also do a reverse study, reverse direction study, and try to see what happens. And that's what the efflux ratios are, without going into the details. And this is kind of a data that you get out of that, and you can interpret based on the rates whether the permeability is good enough or not. And again, how do you know that the permeability is good? Like if you get a permeability of an X value, how do we know it's good? And this is where the historic data really plays into it. Let me see, I'm seeing something here that
Yeah. Yeah.
I don't know what this is actually. Is that a pen or something?
Yeah. I'm just trying to check it out. You please proceed and just make clear that somebody is writing.
Okay, it's not me. Okay, that's good. All right. Okay, sorry for that. So what we're talking here is how do we know what is a good permeability, right? So this is some of the analysis that we have done, and there's a lot of companies do these kind of analysis trying to benchmark their assays. So this is our assay called RRCK permeability. It's more or less like an MDCK permeability screen, and related to the human absorption, assuming that these compounds are not soluble to limited. And when you do that, you can essentially see there is a decent trend here. And you can define a criteria that if the permeability is above 5, it's most likely you are in a very good space. If it is below 5, you probably are not in a good space if you're a very conservative scientist. But if you're very optimistic, you could just argue that I might be right here. Even the permeability is low, but this is where the transporters and other stuff comes into play. But if you just look at the trend, this is what we typically see, right? So based on these criteria, we say is like, if the permeability is more than the specific number, which is 5 in our case, in our assay, we usually quantify that to be a good permeability, and therefore you don't need to go back and try to come in, try to modify the structures and other stuff. So the transporters, it's an important piece. I think there is more and more learnings in this area. It's pretty rapidly evolved in the last two, three decades. And there's a lot of knowledge that's been accumulated now, implying the importance of these mechanisms. It's just a distracting cartoon, but that's how the transporters work, to be honest with you. And there are several types of transporters. I think I don't want to get into the biochemistry of these transporters and how these work at a molecular level. But let's talk more about what are the relevances of these molecular transporters in the oral absorption. PGP is P-glycoprotein, is one of the efflux pumps that are on lots of membranes, including the gut, blood-brain barrier, you name it, like the liver, placental barrier. And the main role of PGP is to push the compound out of the cell. And since it is doing that, if you have a very strong PGP substrate, it could actually PGP can actually limit the absorption of the compound. And this is a demonstration from a PK study with digoxin. Digoxin is a pretty standard PGP substrate. And when you dose it in presence of a PGP inhibitor, which is clarithromycin, you will see a much higher exposure. What that means is in the absence of a PGP inhibitor, you're actually getting a less exposure. your bioavailability is going to be much lower. And the difference is if you age under certain assumptions that clarithromycin is completely knocking out PGP, you can say that this difference is what is driven by PGP, but you have to make those assumptions. So where do we see this often? We see this more in the BCS class 3 and 4. This is a different classification system. And again, I'm not going to touch upon this because we are talking about oral absorption here.
Can you please
Hello?
share your screen? Sir? We have small.
Okay.
Yes.
Okay. Okay, can you see that now?
Yes, sir. Yes, sir. Thank you, sir.
Okay. And should I proceed?
Yes, sir. Please, sir.
Okay, perfect. Thank you. Thanks for doing that. So BCS, that is low permeable compounds, which is class 3 and 4. This is where we see the maximum effect of PGP, and you can see the change in AUC, or the exposure of a substrate drug in the presence of a PGP inhibitor like clarithromycin. You see an increase in the exposure, and that's what you're seeing in case of class 3, class 4 drugs with the PGP inhibition. So if you have a low permeability, which kind of makes sense, isn't it? Like if the permeability is low and you have a substrate to an efflux pump, those are the set of compounds you should worry about the role of efflux and try to dial down that activity if it is feasible. And there are programs which actually play with the structure to try to dial down the PGP activity. But you see there's a lot of those examples from a blood-brain barrier perspective to reach the brain as a target. I think there's a very recent example, a good example from Pfizer. And if you're interested, I highly recommend you take a look at it. It's crizotinib to lorlatinib. Crizotinib is a very successful drug, but it does not get into the blood-brain barrier through the blood-brain barrier, and therefore does not work on the brain tumors. And that's when Pfizer has designed its predecessor, or its successor, sorry, which is a lorlatinib, which does not have the PGP activity. Therefore, it abundantly reaches the brain, and therefore also is very active there, which was only approved last year. So it's a very good story. If just try to remember the names and Google it, you should be able to find that story. Then there's uptake transporters in the gut. These transporters could be very critical based on the chemistry that you're looking into. You don't necessarily need an uptake transporter for all the small molecules. If you have a decent lipophilicity, decent permeability, you don't need them. But if you're again a class 3, class 4, and with a specific chemotype, you probably need it. And there's tons of transporters here. If you look, there's something called OATP2B1. We will talk a little bit about this. Drugs like fexofenadine, celiprolol, some of these statins like pravastatin, rosuvastatin, they use these transporters to actually cross through to get into the plasma. There's other specific transporters like beta-lactams. They use a transporter called PEPT1 to get into the cell and eventually get into the system. Without these transporters, these exposures of these beta-lactams are typically going to be very low. Similarly, there are transporters like commonly used drugs like metformin. There is mechanism driven by the organic cationic transporters, which will facilitate them absorption. So it's important to understand these activities when you particularly have compounds with the low solubility or low absorption, so that see if what you see in vitro can translate into in vivo. It's important for that translation piece. The complexity, however, is high because the expression of these transporters could change as you go across the length of the intestine. For example, if you take ABCB1, which is nothing but PGP, you see the expression increases as you go from duodenum to the ileum, and then it drops suddenly in the colon, right? So if you have a strong PGP substrate, your absorption may be limited in the ileum piece, which could be important because it's one of the functionally active absorption site. The other transporters, like the other example, PEPT1, also is highly expressed from duodenum to ileum, but you don't see anything in the colon. And this might be important when you're thinking about something like entericoding or the high dose drugs, because once they hit the colon, you won't see anything absorbing for those guys. So understanding these expression levels and the functional activity of those proteins in these different segments is pretty critical when you're dealing with those transporters with those kind of chemical spaces. And as I said, the complexity increases because these expression levels could vary based on the site of the gut. I'll talk a little bit about OATP2B1. We believe this is an important transporter for a lot of acidic drugs, like a lot of statins are being absorbed through this mechanism. And we definitely have evidence, clinical evidence to support that. The good part about 2B1 is expressed throughout the gut. So even if it is absorbed slowly, it can still be absorbed in the colon, therefore get a highest activity or the overall absorption as possible. So fexofenadine is a very good case example when we talk about the role of intestinal transporters and sometimes think about how complex it can get, right? Some of you might have known the story of terfenadine, which was the first drug in this space, or which was the first molecule which was approved, which is Seldane. This is again actually the molecule from I think it is Wyeth. Maybe somebody has to correct me if I'm wrong there. But anyway, terfenadine, because it's of very high oxidation, it converts into fexofenadine, and fexofenadine is the molecule which is actually active molecule. So eventually this got withdrawn, and there was development of fexofenadine, which is now Allegra, which is available everywhere. Allegra, which is fexofenadine, is a carboxylic acid, and it utilizes several transporters in order to go from the gut lumen into the blood compartment. And those transporters include OATP2B1. It's also very strong substrate to PGP, which means although this is pumping in, this is pumping out. So based on the concentrations in the lumen versus the cellular concentrations, one of these will predominate and have the overall effect. So this plot, I don't know if you can see all of that. So this plot is demonstrating that the effect of apple juice it actually inhibits OATP2B1, and if you take apple juice with fexofenadine or like orange juice or grapefruit juice or something like that, if you take it with fexofenadine, you technically get no exposure and you don't get relieved from the allergies that it is supposed to treat. Therefore, it's clearly no to take some of these drugs with the juices, and this is the primary reason why you should not be taking with juices, because you're essentially inhibiting this transport mechanism at the gut. So again, there's some very successful drugs out there in the market which efficiently uses these transport mechanisms. Some of the good case examples are the Pfizer drugs pregabalin and gabapentin. So pregabalin and gabapentin uses an amino acid transport system, which is some of these amino acid transporters like B0 and BO positive. And we don't really know the contribution of some of these variants, but we definitely know that these mechanisms contribute to the overall activity. So pregabalin and gabapentin use these mechanisms to get into the enterocytes. And once they are in the enterocytes, they use a mechanism called LAT2 to get into the blood compartment. And once they're in the blood compartment, they have to go through a LAT1 transport mechanism in order to go to the blood-brain barrier, which is the site of action for these molecules, right? So technically, we rely heavily on the transport mechanisms to cross each and every barrier to reach the site of activity. Uh, and if you're working in a space like this, it's obviously, you know, how important is it to really understand these mechanisms and try to quantify that in the process of translation. I think so the other story about pregabalin is, sorry, the gabapentin is because of the limitation of the transporters in the small intestines and not present in the colon. We see a dose-dependent absorption. That means at the highest doses, the absorption, overall absorption is low. There are groups which actually looked into the prodrug approaches to target other transporters and essentially reduce, limit that absorption site, and have the absorption happening all through the small intestine and the colon. And this is the example of that. It's a very good story of gabapentin and Cabrol. It's essentially a prodrug which uses a different transporter than what gabapentin uses, which is MCT and SMBT, which is a sodium multivitamin transporter. And these are efficient enough to actually carry even the highest dose across the gut membrane. So these are just some of the examples to demonstrate how these multiple components play a big role. And again, there's a whole set of tools we can utilize depending on the stage of your discovery development, and try to characterize these mechanisms, the passive permeability, the transport mechanism. And then we also talked about some of the fundamental tools that you use for solubility assessments and other stuff. And technically try to create a map of all this information to feed into some kind of a mechanistic model to better understand what is happening. So I'm only left with a couple of slides, which is good. So I think we are right on time here. As I said, gut is one of a complex organ from a drug disposition point of view. That's because it has very different anatomy and physiology as we go from one end to the other end. And these individual processes of solubility, disintegration, dissolution, and membrane permeability is happening across these segments, and they could vary. These processes could vary because of pH differences and other stuff that we talked. And it's important to integrate all this information from a different segment in a more mechanistic sense. And this is where the physiologically based pharmacokinetic modeling really helps. It's trying to use that fundamental property details of a molecule, which is like the solubility, permeability, and all the phys-chem properties, and try to translate that information at an individual segmental level, and then eventually talk about the contribution to the FA. And it's when you know nothing about the compound except for with the animal data, this kind of information is pretty critical in translating your in vitro data to predict what is going to happen in the human when you dose it for the first time. And this is the final slide. There is a lot of exciting work going on, trying to, some of the technology groups trying to come up with the artificial systems. Like this is an example of an artificial gut, which essentially have these multiple components of, for example, the stomach component. And from the stomach, it goes to the jejunum component, and there is a solubility compartment. And for each levels, there is a permeability compartment and stuff. And this is a complex and a dynamic model. But these models there's a lot of activity in this space, like the microphysiological systems and other stuff. And this could definitely be one of the promising areas as we try to understand these complex mechanisms as we move forward. And this is obviously, there's a lot of work that needs to be done here. probably a lot of opportunities for scientists who are trying to invest into these areas. That's about it. I know I don't have a conclusion slide, but I think I tried to tie the whole story together, how things are being considered in a drug discovery setting, to going towards the FIH, and what are the multiple components in this, and how do we go about it. And I will be very happy to take any questions. I really thank you for your attention. Yeah, I'll stop there, Ravi. Dr. Ravi Kumar.
Sorry, sir. We have some technical issue. Give us 2 minutes, sir. Ravi sir stuck on technical issue. Am I audible, sir? Verma sir?
Yeah, I can hear you very clear. Thanks.
Yeah, 1 minute, sir. There is some technical issue. I'll solve it.
Yeah. Can you hear me, sir? Dr. Verma?
Yes, yes, Dr. Ravi.
Thank you so much. It's an excellent presentation. Slides are really beautifully made. So all our pictures and pictorial diagrams, the concepts are very clearly, especially the pharmacokinetic modeling and the deep core concepts given excellent. Presentation.
Thank you so much. You know, my goal was to give an overview. There are a lot of intricate details, and definitely there are experts in this call. I'm very happy to have any follow-up questions through this chat or even through the emails. Happy to address some of those.
Yeah, with your permission, can we go to short Q&A session for just
Yes,
5 to 10
please.
minutes? Yeah.
Yes, please.
So, yeah, now I request audience, participants through Zoom, if you have any questions, please raise your hands and unmute yourself and directly ask the speaker. So meanwhile, I have few questions with me. So shall I go ahead, sir?
Please go ahead.
So throughout your presentation, I've seen that most of the major focus is given to improve the bioavailability. So if that is the case, then what would be the efficacy of the drug? So can we balance the drug efficacy and bioavailability, because there are several methods which you employed, I mean the companies are employing, that is the structural modifications. You have shown some drugs also. During that time, how can we balance that the efficacy of the drug is not altered?
Right. So yeah, that's definitely one of the major components in the drug design, right? You're looking at a molecule from a different angle. An ADME scientist is looking from an exposure point of view. Then there is an pharmacology group which is looking the same molecule from an efficacy point of view. So when you're optimizing this molecule, we need to really find that sweet spot which has all these activities, and obviously that's a very important component of trying to optimize all of these simultaneously. We cannot compromise on the pharmacology just to increase the exposure, because over there and your end product is not changing. So yeah, you're absolutely right. We have to optimize, and we do optimize all of them simultaneously.
Right. So I have one more question, that is, so these ADME optimization models you present today mostly on NCEs, especially for the oral absorption drugs. So these concepts which are working, can be extrapolated to other routes of administration, some of those models?
I think to some degree, I think it would work to some degree. Obviously, the solubility concepts and the permeability concepts may not be different across different barriers, although you probably need to consider some of the physiological components. For example, the nozzle has a different different environment, different level of water available, different pH there. So once you account for all of that, the fundamental components will still be the same. The fundamental intrinsic solubility and intrinsic permeability is going to be more or less the same. I did not really talk about the MEE part of this. I only spoke about the A of the ADME part. Obviously, the metabolism, disposition, and elimination, they are going to be the same irrespective of the route you do, because most of this disposition is happening because of liver transport or metabolism or maybe renal disposition. So those will definitely remain constant.
Thank you, sir. Ramarao, sir, would you ask something to the speaker, sir? It seems you are waiting to ask something. Ramarao, sir?
Oh, Dr. Mantha, Ramarao here.
Dr. Rao, sir, how are you?
How are you?
Long
I was
time.
busy.
I'm very excited to see you. Actually, I was not expecting.
Thank you, thank you. One simple question.
Mm-hmm.
You are actually talking about different animal models.
Okay.
There are a lot of actually the differences. Jejunum is not there in all the species. Say, for example, one simple point.
Yes.
And the enzyme systems, whatever you talk about transporters, are there any comparative studies? That is what I was looking at it.
Yeah. So yes, first of all, as a philosophy in Pfizer and a lot of other large pharmas, we try to follow the RRR, right? The triple R, try to reduce the animal usage and other stuff. So pretty much in all these slides, I did not show any animal data because we don't really use a lot of that data. We predominantly rely on the in vitro data, cell culture data, and the in vitro data. That said, there is always some sort of an animal information that we gather, which is predominantly the rat absorption as well as the dog absorption, because that's some of those requirements for the BCS purposes and other stuff. And you're absolutely right. There is species difference in the physiology, in these protein expressions, and other stuff. So it gives you a ballpark, I would say. It does not give you an accurate translation. But again, it's like trying to do an orthogonal prediction, right? Like if your rat data shows high absorption and your in vitro data also shows very high absorption, I think you're in a decent space to move forward with that, right? So it may not be necessarily quantitative because of those differences, but at least it gives you a direction of whether it's going to be successful or not.
Yeah, as long as there are no actually qualitative differences, quantitative differences is fine.
Yeah.
In some species, the transporters are absent. If I am correct. So that actually the predictions, are these transporters throughout any study is there? That's what I was looking at it.
Yeah, there is data on the expression of, let's say, rat intestine versus human intestine versus monkey. There is data.
Yeah,
is there a functional data? there is not an extensive functional data to say that what you're seeing in the expression really translates into it. There is definitely a gap there. But as you said, there is definitely differences in the proteins that are expressed. For example, if rat expresses OATP, if human expresses OATP 1B1, rat instead expresses OATP 1B2, which is not present in the human, right? So there are definitely those species differences.
Yeah, that is not, yeah, that is more important, because even actually even human genetics also variable. So these transporters are differently present.
Yeah.
So automatic.
And as I showed in some of the slides, it's not just these transport proteins, but the physiology changes, the pH changes, the luminal activity changes. I did not touch upon the gut microflora. That's a whole another topic that is very dramatically different and as the age group goes on. So yes, totally agree.
Yes, that is perfectly correct, because their food habits also makes a lot of difference.
Right.
Carnivorous versus herbivorous animals, the digestive tract system is entirely different. Anyway, that is one questions which are coming up. Otherwise, theoretical only. Practically, I do not know theoretical aspect. I just
Yeah,
ask.
we definitely look into that stuff. To some degree, obviously, it's not an academic institute, so our goal is ultimately to mush the molecule, not necessarily to understand all the molecular basis of it.
Yes, that's correct. That's correct. Good, good, good. Nice actually interacting with you. Good aspect.
Thank you so much.
Yeah, yeah.
Good seeing you.
Yeah, sure. Thank
So much,
you.
Ramarao sir, for your interaction with the speaker.
Thank you.
Sir, one last question I have. There is a 5-thumb rule you mentioned about permeability. 5-thumb rule. Do you hear me, sir?
Okay. Yeah,
So
yes, I can hear you.
do we have any close relation with the BCS classification? That means this rule can be applicable to all the classes of BCS, or any one particular class is more relation to this rule?
I think they are very related. I think if you think about a big picture, what BCS tells is more or less what rule of 5 tells. For example, if BCS class 1, which is high solubility, high permeability, which means complete absorption, is pretty much a compound with rule of 5 of no violations. So they will technically fit in the same bucket if you take this set of compounds, put it in that grid, or take that compounds and put it in the rule of 5. In a big picture, they should give you a same answer, but obviously there's always outliers.
Thank you so much, sir. It would be very interactive, but limitation in interest of time, we can close the Q&A session. So I'll request Dr. Nagarjuna Reddy, Associate Professor, Sri Vishnu College of Pharmacy, to give a formal vote of thanks now.
Thank you, sir. Thank you, Honorable. On behalf of Sri Vishnu College of Pharmacy and entire fraternity of management, it's my privilege to propose vote of thanks for Honorable Speaker of this section, Dr. Varma Mehta Garu. In spite of your busy schedule, sir, we have given a valuable speech to our participants and also for us to learn something. It is always interesting to learn such scientific talks on the proposed topics like gut instinct, understanding oral absorption of your molecules. I express my sincere thanks to our director, sir, Kumar sir, Director Kumar V.S. Nemmani sir, for arranging today's session, and also for about this topic to be delivered. Thank you, sir. Thank you, Director sir, for giving this opportunity for us to learn, and also I will thank Principal Sir Dr. K.S. Natarajan Garu for his support to us to conduct the section in this smooth and manner way. Extend my thanks to all my coordinators who are working off the screen, especially Mr. V. Ramakrishna and Dr. Bhagiraj. Last but not least, I take the opportunity to thank our Chairman Sir, Dr. K.V. Vishnuraj Garu, and also our Vice Chairman, Dr. Ravichandran Raj Gopal Garu, and Secretary for Sri Vishnu Educational Society, Sri Aditya Visham Garu, for providing this support. At this juncture, I also thank all participants for your extended cooperation and hope you have acquired significant knowledge for this session. Thank you, one and all.
Thank you so much. I'm going to sign off, and I really appreciate the opportunity to interact. And I thank you, Dr. Reddy, Dr. Ravi Kumar, and Dr. Nimani. I hope to interact again. Hopefully, when I come to India, maybe I'll stop by, because I always visit East Godavari, West Godavari.
You are always welcome, sir.